Today I'm going to talk to you about one prong of research in my lab. And for those of you who are very genomically based, I'm going to disappoint you straight away. There is just one FST plot in this whole talk, right? So for the most part, I'm going to be talking about phenotypes and some of the underlying potential genotypes that we see. And this prong of research in my lab is about thermal effects on reproduction. So I'm very interested in the evolution of reproductive strategies. We all know that climate change is happening. We have an increasing in the mean and variability of temperature. So the number of heat waves, their intensity, their length is increasing. And in response, organisms have either shifted their ranges poleward or they've changed aspects of their phenology to be, for example, reproducing earlier in the spring. But I'm also interested in what other traits might be important in thinking about responses to increasing temperatures and how this might affect some of these responses that we're seeing, like shifting ranges, for example. A few years ago, some collaborators of mine, we published this tree paper on the impact of climate change on fertility. And we were interested in this subject because most of the predictions about how organisms are going to respond to future climate change is based on critical thermal limits. That is, how hot it needs to get before organisms die. But in the literature, you'll see comments that reproduction seems to be typically more thermally sensitive than survival traits, meaning that it doesn't have to get as hot before effectively you lose reproductive function. So you're surviving, but you're not having any offspring. And many reproductive processes can be negatively impacted by thermal stress from, I have to do this so everybody on Zoom can see it. From gametogenesis, making gametes, all the way to finding partners, courting partners, and exchanging gametes, to fertilization, to offspring development. And so for the past few years, my lab has been focused on the impact of thermal stress on fertility and its potential evolutionary and ecological consequences. I'm one of the co-leaders of a European Society for Evolutionary Biology special topic networks on the evolutionary ecology of thermal fertility limits. And we recently published a paper talking about methods in which one could assess how fertility is affected by high temperature stress. So here what I'm showing is your typical thermal performance curve where you have some performance trait like heat knockdown or some aspect of locomotion, and you're measuring individuals across a different range of ambient or ectotherm's body temperature, and you're trying to determine the minimum temperature at which movement stops, the maximum temperature at which movement stops, and of course then you can also characterize other aspects of this thermal curve. And because you're measuring different organisms the same way, then you can compare these values across different taxa. Another way to get the same kind of data is instead to do a lethal limits approach where instead of some proxy for fitness, some performance trait like locomotion, you're actually measuring survival. So here you have survival and you expose different individuals to different temperatures, and then you can measure the temperature at which 50% of the population don't survive that heat stress, so that's LT50, or 80% of the population doesn't survive that heat stress, LT80. And we suggested that actually you can apply these same kinds of concepts to thinking about how fertility is affected. So here we have a thermal fertility limit, or TFL, and instead of measuring survival, you're measuring fertility. So these are also 0-1 traits. So here you have fertility and you're exposing different individuals to increasing temperatures, and you can measure the temperature at which 50% of the population is sterile. And by that, I mean they either produce one or more offspring or they produce none. So the amount of offspring that they produce is not important, it's just 0-1. Or many studies take what we call kind of a sensitivity approach, where it's not quite the thermal performance curve, but it's not a limits curve either where you expose a subset of the population to a benign temperature and a subset of the population to a hot temperature, and you measure some difference in a quantitative trait. For example, here, fecundity, very close to fitness. Okay. Okay. So you might be thinking, well, what's new in all of this? Because we know that we have some ideas that the thermal sensitivity of fertility is well known in, for example, plants, Drosophila, and, for example, mammals, primarily because of the interest in the agricultural field. It's also thought that male function is typically more thermally sensitive than female function. But what's new in this kind of research realm is its application to understanding climate change and how this might influence our predictions about what's going to happen in the future. So as part of this STN, we also recently published a systematic map looking at literature that addresses how temperature influences reproductive traits. So we searched the literature for published papers testing the effect of at least 2 temperatures on reproduction. By reproduction, we mean fertility, fecundity, mating number, gamete traits, gonad traits, or, for example, the proportion of individuals breeding. We didn't look at courtship traits, for example, so it was just stuff about whether reproduction was happening or not. We then categorized these studies based on taxa, the reproductive traits that they measured, things like whether they were ectotherms or endotherms, reproductive mode, sexual, asexual, fertilization mode, external, internal, etc. We ended up screening close to 15,000 papers. This could only be done with a group of that size. After looking at all those papers for a number of criteria, we ended up with over 2,000 texts that we actually looked at fully, and we included over 16,000 in our map that represented nearly, 1,600, representing nearly 1,200 species. I'll answer it that way. Here's the taxonomic distribution of what we found. We have 12 animal phyla represented, but 65% of them are arthropods, and of those, nearly 50% of the studies were based on insects. And amongst those, even insect taxa are not equally distributed, as one might predict. Most of the highest number of studies were done on Drosophila melanogaster, and I'm going to show you a couple more as we go through. But we can also think about of the organisms that were most represented in these studies. Besides melanogaster, we have the spotted winged Drosophila, so a pest species, red flower beetle pest, diamondback moth pest, aphid pest, codling moth pest. And we have the honeybee, pollinator, and a disease vector. Right. So there's also a lot of interest in the effects of temperature on, for example, pests and pollinators. By far, fecundity was the most common trait measured. Fertility was rather low. So most studies assessed either offspring or egg production. That's how we categorize fecundity. Interestingly, most studies simultaneously exposed both males and females. This is a problem because then you cannot discriminate how thermal stress affects male reproduction and how it affects female reproduction. Most studies use chronic thermal stress. And here we're saying chronic being 5 days or more. Now, obviously, that depends on the lifespan of the organism that we're talking about. This is kind of more like a heat wave type of effect. Most studies are exposing adults to these temperatures, and most studies are using constant temperatures, despite the fact that we know that thermal environments are much more complicated through fluctuating temperatures. So that gives us an idea of the distribution of studies that are currently out there and maybe a focus for future questions to start to tease apart how thermal stress is actually affecting reproduction in populations. So some of the questions I'm going to talk to you about today is fertility actually more sensitive than survival? Are males more sensitive than females? What's the effect of constant versus fluctuating temperature on this pattern? Is there any adaptive potential to respond? And if there is, we might want to know what the underlying genes are to that, because then we can look at other populations or other species and see if we can find some similar patterns. And importantly, if all of this is going on, what are the potential consequences on population fitness and downstream effects? For example, like its effect on mating systems, sexual selection, things like that, that can influence population dynamics and the viability of populations over time. Okay, so again, as part of a group, we were trying to answer the first question, which is, does sterility occur at lower temperatures than death? Because our whole idea of these thermal fertility limits is that they're shifted to the left of survival limits. That is, they're more thermally sensitive. So we tested 43 Drosophila species, these are listed here, where we exposed only males, so we didn't look at females at all, to a 4-hour adult. Okay, so again, we're kind of repeating some of the problems that we found in our systematic map, but a 4-hour adult heat shock across these different temperatures here. And we measured two things, survival, and if individuals survived at that temperature, we then took those heat-treated males, mated them to non-heat treated females, and then assessed their fertility. And again, it's just 0, 1. What you're going to see, you can see that these are not arranged phylogenetically. They're arranged by thermal sensitivity. In the next slide, you're going to see for each species 2 dots. A black dot, which represents the lethal limit, 80%. So 80% of the population dies after exposure to this temperature. The pink dot is the TFL, the thermal fertility limit. 80% of individuals that's are sterile. And here's what we find. So we can see that for some species, the pink dots are indeed shifted to the left of the black dots. So sensitivity, fertility is more sensitive than survival. But that's not true for all of the species. Overall, 44% of the species had TFLs that were lower than either our calculated lethal limits, and I'm not showing you the data here, but also we calculated the critical thermal limit where nothing was happening. And for 44% of those species, there was a gap between the survival limit and the fertility limit. The average difference was 1.15 degrees Celsius, but it ranged from 0, where there was no difference between the survival limit and the fertility limit, to up to 4.3 degrees difference in the sensitivity of these 2 different traits. And right now it's ranked by robustness for survival, but if we were to reorganize this for robustness for fertility, it would change the rank order of these species. We looked at the correlation between these values, and we do see that these 3 values, the thermal fertility limit, the lethal limit, and the CT max, are all correlated, but the 2 survival measures are only weakly correlated to the fertility measure. So this means that you can't really confidently use the survival measures to predict the fertility costs. We also tried to look at any ecological factor that could explain this variation, but there wasn't any ecological factor that we could associate with this pattern. Okay, so that's all right. Now we've shown, well, in some of these species, the fertility is more sensitive than survival, but what's the evolutionary or ecological consequences of this? And what we found is that these TFLs, these fertility limits, better predict current species ranges than CT max or lethal limits. And those are the values that people have been using to predict what's going to happen in the future with species. So if you're using those values, and for that particular species, their fertility sensitivity is at a lower temperature, then you're really underestimating the potential vulnerability of that species to future climate change. Because we also found that if you use these fertility limits, species are generally tending to live closer to their thermal safety margins than if you were to use the survival limits. Okay, on presentation mode. Okay, so if TFLs better predict species distributions than CTLs, and we want to see how that's going to play out over the future, we did this model simulation. I don't want to get into that. We looked at the current climate and we chose Drosophila flava montana. It does have one of the biggest differences between its survival temperature and its fertility temperature, but we also chose that because its distribution is very well known. It's just restricted to the Western United States, and it's been sampled where all of these X's are. And this represents the current climate, and the blue is the potential niche based on CT max for this species. But if we add places where it's too warm to reproduce, that's the orange. Well, we don't actually see any living in there, and the too warm to live is the red. So you combine the red because obviously that's already hotter than what they could reproduce in. So this was a while ago, so we're using the old IPCC models. So here we're using RCP 4.5, which is an intermediate. Let's try and keep warming down to about 2.5. And you can see that the species loses habitat primarily through loss of suitable habitat because now it's too hot for them to reproduce. This is the worst case scenario, so we do absolutely nothing and we go about 4. And you can see that we lose a lot of habitat. And again, it's more limited habitat suitable for reproduction compared to survival. And because these TFLs do a better job of predicting up to increasing the quality of the predictions, the accuracy of the predictions by 35%. So I'm not talking just a little increase in the predictability. It's a huge increase in the predictability. There must be some ecological relevance to this. Simultaneously to this experiment, another group in Australia was doing something very similar, asking very similar questions, but what's the effect of increasing temperature on population extinction, and what is the influence of male and female thermal fertility limits on this process? And also asking, is there any adaptive potential for populations to respond to this? They looked at 6 species, and they put them in an experimental thermal evolution situation where they started all 6 species at 26 plus or minus 3 diurnal fluctuation, and then overlapping generations, and then every 2 weeks they kicked up the temperature by 0.2 degrees. So fairly rapid increase in temperature, and they continued to do that, and they were measuring census population size. And obviously, when those dots get to 0, the population is extinct. And you can see that many of the populations go extinct quite early in the process. And that there's a difference. The dotted lines are tropical species and the solid lines are temperate species. Now you might say, okay, well, what does this have to do with fertility limits? It's getting hot, so they're going extinct because they can't survive. So they also quantified, similarly like we did, the male fertility limit, but they also did the female fertility limit, CT max, and the lethal limit. And then they looked for the relationship between those values for each of those 6 species and the average extinction temperature. And both the male fertility limit and CT max had greater predictability than the other the lethal limit, or even the female fertility limit. And you can see that the female fertility limit is generally occurring at a higher temperature than the male fertility limit. So this suggests that males indeed are more thermally sensitive than females are, but they also found the same as we did. Thermal fertility limits were better predictors of current species distributions. Tropical species were more sensitive than temperate. And because they never saw any either stabilizing effect on the census population size or increase in the census population size, they argued that there was no adaptive potential. Okay, 0.2 degrees every 2 weeks, kind of tough. But what I think is really interesting about these two studies is they approach this problem in two very different ways. We employed heat shock. They employed chronic thermal stress throughout all developmental stages. And we see the exact same thing. These TFLs better predict current species distributions. And they put populations at risk, and it's probably driven by heat-induced male sterility, with maybe limited adaptive potential to respond to this. So as evolutionary biologists, a major challenge for us is trying to understand the relationship between variation in quantitative traits, selection, and the underlying genetic variation. But that's really critical for understanding how populations and species are going to respond to climate change. So my lab then started to look at the evolutionary potential and other aspects related to these thermal fertility limits. And so we have started to look at whether there was any within population variation in sensitivity of fertility to thermal stress. And if there was, what's the heritability of that? And what's the underlying genetic basis? And to what extent actually are males and females of the same species differentially affected? So to do that, we used Drosophila melanogaster. So I'm adding to that top row. And we used the DGRP line, which is a series of over 200 isofemale inbred, nearly isogenic, not quite entirely, lines from one population in the east of the United States. And we exposed 127 of these lines to 3 different developmental temperatures, both males and females, although not, you'll see in a minute. We then took these treated individuals and we mated them to control individuals of their same line. And then we recorded fertility, again, 0 or 1. I'm going to show you the male data here. So here's a proportion of individuals in the population for each of these lines that are fertile at these 3 different temperatures that we used. And when we do our statistical analysis, the proportion of fertility is highest at 25. It significantly decreases at 27, but it really decreases at 29. But the interesting thing to me about this is that some lines maintain their thermal robustness and other lines are starting to tank. So we calculated broad sense heritability because that's all you can do with these lines in the way that we use them. And we see that at 25, our heritability is 0.18, but at 29, because thermal stress releases cryptic genetic variation, we see that broad sense heritability goes up to 0.4. Whether that could result in an evolutionary and adaptive response in the future, we don't know, but at least there's a little bit more genetic variation to act on. Now, the nice thing about these DGRP lines is not only are they sequenced, but they've been used a lot by other people to measure traits or trait response under different stress, desiccation resistance, oxidative stress resistance, lots of other things. So we took data from CT-min, CT-max. We took data from all of these other studies where we used the same lines and we looked for correlations. And the only correlation that we found between thermal fertility sensitivity of these lines was the effect of oxidative stress on lifespan. So the less sensitive lines, these guys up here, also tended to live longer after oxidative stress exposure. The more thermally sensitive lines lived less long after oxidative stress exposure. So oxidative stress will come up again. So remember that. Now remember, I also said that we looked at females, but we didn't look at all of the females because we had already had some preliminary evidence that suggested that females weren't as sensitive. So we took 20 of the top performing male lines and 20 of the top performing, or poor performing male lines, and we looked at the female effect. And 25 and 27, there's no significant difference in line fertility, but we do see a drop in fertility at 29, but clearly it's not as severe. And more importantly, male reproductive performance doesn't predict female reproductive performance. So here we're saying males are more thermally sensitive than females. Okay, but that was constant temperature. What about a somewhat more realistic fluctuating temperature? So we did the same experiment, except now we're just focusing on these 20 high-performing lines in the black dot and these 20 low-performing lines in the gray dots. Here's our constant results for fertility. And then we also did this fluctuating experiment where we had the same average temperature, but for 25, we went up 4 and down 4. And for the 29, we went up 4 and down 4. So that this means the 25 fluctuating temperature spent part of its time at 29, but then also part of its time at 21. And the 29 treatment spent part of its time up at 33, but part of its time at 25. So there was some overlap in the high temperatures and the lower temperatures. And what you can see here is that fluctuating around 29 makes all males worse. Even the high performing lines, they started to fail in their fertility measures. But the low performing lines completely tanked, absolutely completely tanked. Now one could say, well, if these low performing lines, maybe they're just shit, right? They're just crappy all the way around. But what we also measured were some other traits, such as mating success. And you can see here that either under constant or fluctuating, we don't see clustering of the high performance or the low performance lines. And that we can see under constant, the lines don't have any particular pattern. Some lines actually increase their mating success at 29 constant, some decrease. But when we move to a fluctuating system, almost all of the lines decrease in their mating success under high fluctuating temperatures. So not all reproductive traits are responding the same in response to temperature. Okay, so there's your FST plot. Look at it, enjoy it, because you're not going to see anything genomic again. So we took these 20 high and these 20 low lines, and because they had been sequenced, we performed an FST outlier analysis, and we found a bunch of SNPs, because there's a bunch of SNPs that differ between these lines, but we only found 54 that significantly associated with the phenotype. We then took those 54 and we went 2 kb up and downstream, and we found 16 genes, and 11 of those were on the X chromosome. Now, this effect is probably an underrepresentation because the DGRP is now sequenced for the Y chromosome. And we already know that male fertility genes and melanogaster are on the Y, some of them at least. So this is probably an underrepresentation of actually what's going on. Okay, so as I mentioned, the effects of heat on mammalian fertility are well studied because of the agricultural implications. And this review here kind of summarized the effects of scrotal heat tests. I showed you that picture early on of the red bag heating up a male scrotum. And they looked at the processes that are negatively affected by this. And they all essentially lead to apoptosis and autophagy through altered gene expression, DNA damage, stress response, impaired DNA repair, triggering this apoptosis pathway. And sterility can be due to problems during spermatogenesis. And spermatogenesis is actually a fairly conservative process. So we might then expect to see similar problems in melanogaster as we do with mammals. So we have those genes. What did we find? So because it's melanogaster, there's good annotation, some of which are used for based on mutants and things like that. And when we could find the function of at least one of the functions of those genes, we could put them into three general classes of candidate genes. Those affecting male reproduction, those related to oxidative stress and DNA damage, and those related to autophagy. So for example, our genes that we put in the male reproduction category were known already to negatively impact male fertility across both plants and animals. Because spermatogenesis, doesn't matter whether you're doing it in pollen or whether you're doing it in sperm, it's very conserved. Regulates fertility and represses expression of heat shock inducible genes. So that kind of DNA repair process. They're involved in morphological defects in the melanogaster testes, play a role in the CAMP signaling pathway in Drosophila spermatogenesis, which is important to make sperm. The oxidative stress and DNA damage genes. Mutants are known to be hypersensitive to oxidative stress. So that might be why we're getting that positive correlation with oxidative stress and have decreased DNA repair ability. And we have several genes that were in the autophagy pathway, and autophagy activity increases under stress, and mutants of these genes have reduced activity that is known to impact male fertility through the loss of early cyst sperm cells. So we have, I think, a reasonable list of candidate genes. Okay, but that's where we are with that little bit of the story. So across Drosophila, we've seen that these thermal fertility limits have important and underappreciated evolutionary and ecological consequences. They're associated with range limitations. They can predict extinction. Within a species, there appears to be genetic variation and at least some broad sense heritability for the heat-induced male sterility, not female sterility. And this is putatively underlain by genes associated with impaired sperm function. But as I said at the start, I'm very interested in reproductive strategies. So assuming then that this is a relatively common problem of heat-induced male sterility, I'm also then interested in what are the other consequences of this process. So for example, temperature effects on fertility are going to alter the functional operational sex ratio. So if you have males that are effectively, while they're mating, but they're not generating any progeny, then the operational sex ratio is going to shift to be female bias. That's going to alter the strength and the operation of sexual selection. Temperature effects on fertility and fecundity obviously depress census population size, and that's going to alter other aspects of sexual selection. So, for example, like sperm depletion. Those few males that are resistant or that escape heat stress are going to be quite in demand by females. And both of these things are going to affect the effective population size, and they may alter the mating system. So this one process could have quite a lot of downstream consequences on a species' population dynamics. So to start to address this, we switched Drosophila species. Don't find melanogaster that interesting. And we started working on Drosophila subobscura. And the reason for that is most mating systems are polygamous. Males mate with multiple females, females mate with multiple males, but not subobscura. Females are resolutely monogamous, except when they aren't, and they aren't when they get a dud ejaculate. Some females, sometime, will remate if their first mating is non-productive. But also, interestingly, males provide nuptial gifts. males vomit up crop contents and feed them to females. And previous work in this species by Alina Immonen used the species and manipulated dietary stress to look at the costs and consequences of gift production, acceptance, and use. And so she did this properly where she looked at male effect and female effect. So she has well-fed males and females, starved males and females, and then the reciprocal process here. So starved females, well-fed females. I'm not showing you the male data, but starved males produce fewer drops, fewer are accepted by females, and these males have reduced mating success because females have to accept the drop to mate. Starved females will increase their offspring production to unstarved females, but only when they mate with a healthy, well-fed male. When they're mated to a starved male, they suffer fitness consequences. Okay. But nobody had looked at temperature effects on this process. So we decided to do that. And we were also interested in this species because work in my lab had previously shown population variation in heat-induced male sterility that varied across latitude. So lower latitude populations were more resilient to heat stress than higher latitude populations. And in those higher latitude populations, 23.5 degrees C rendered about 50% of the males sterile through loss of sperm motility. That's been repeated by other researchers in other populations of subobscura. So this is a very common response. But I was interested in asking within a population, is there genetic variation for the effect of heat stress on fertility? And also, is there genetic variation for the propensity for females to remate when they receive a dead ejaculate? Because if there is that genetic variation, then shifting the mating system from monandry to polyandry could potentially rescue the population from the effects of heat-induced male sterility. Okay, so to do that, we performed two experiments using 14 genetic lines from a population of subobscura collected in Edinburgh. And for male sterility, we exposed them to 3 different temperatures: the benign, the intermediate stress, and the really stressful. I will show you data in a minute, but this results in almost everybody being sterile. And we took the males that are closed from these treatments and also the females from the benign treatment. And we mated these benign females to males of each of the types. We put those females in our fertility assay and tested for the effect of that. For female remating, we only used these 2 treatments because we wanted our males to be pretty sterile. So we wanted to see how females would respond to that. So again, we collected males from both treatments and the control benign females, and we mated those females to a male of each type. And we put females in a vial for 4 days. And then 4 days later, we offered her the opportunity to remate with a benign male. And then regardless of whether she mated or not, we remated, we put her in a vial for 4 days. And we counted fecundity, progeny production. Okay, what did we find? So here's male sterility at 18, 23.5, and 25. I don't show it in this plot, but the slopes of the lines for all of the, between the temperatures for all the lines do not differ. So there's no genetic variation in plasticity of this trait, but there is genetic variation for the probability of sterility, but only at 23.5. At 18 and 25, everybody's either mostly fertile or everybody's mostly sterile. And so there's no broad sense heritability at those temperatures. Again, whether this could lead to an adaptive response or not, we don't know. Okay, so what about female remating? So Here's whether the female's first mate was benign, where most of the males are fertile, or with a 25-degree male, where most of them are sterile. And the probability of remating significantly increases when they first mated with a sterile male. Here you can see the lines. There's actually, again, genetic variation for plasticity is non-significant, but we do see genetic variation for the probability of remating at 25. So again, if this converts to additive genetic variation, then we might see a response. Okay. We also looked at what happened to females once they were either singly mated or mated again. So here we have females who only mated once and females who remated. And their first mate was either with a hopefully fertile 18-degree male or a sterile 25-degree male. And here's the number of offspring in the first vial, the second vial, and total. And what you can see here is that monogamous females mated to 18-degree males have the highest fitness over any of our other categories. Females who first mated with a sterile male produce no progeny in the first vial, but do start obviously producing progeny when they remate, but obviously that amount of progeny does not compensate for that first 4 days that they missed because they mated with a crappy male. I'm running out of time, so I'm not going to talk about those two. They're interesting, but they're a little complicated. We also measured survival probability. And you can see here that if a female first mated with a 25-degree male, she died faster than if she mated with a normal male. So not only does she not have any progeny, but insult to injury, she dies earlier. Okay, now what about that vomit? Let me show this to you. Okay, so this is a female, this is a male. He's working his way up to this spitball. He gives it to the female, she accepts it. She's like, no, not gonna mate with you. So very frequently males have to give females more than one nuptial gift. So he's working up to another one here, but then nothing happens for a really long time. So I skipped ahead for you all. Here he's doing another one. You can see that this is kind of not easy for the male to do this. She accepts it. And this time she mates with him. Okay, so to look at how heat stress affects male gift giving behavior and male gift and gift acceptance by females, we did a very similar experiment, only this time we used, sorry, don't yell at me, only 4 lines because it's really looking at a lot of videos, it's not very fun. High and low temperature. We take individuals for those and we put them in low with low, high with high, in the reciprocal cross. And we measure things like the probability that a male gives a gift. And if he does give a gift, how long does it take? And the number of gifts presented. We look at, well, if a male does present a gift, what's the probability of the female presenting, of accepting it, and how long does it take her to accept it, and how many gifts does she accept before she's willing to mate, and then what the fitness consequences of that is. You're almost done. It's vomit, hey. Okay, so here's gift presentation probability, gift presentation latency, and the number of gifts presented by males from the benign versus the heat treated when they were mating or interacting with females that were from the 18 or females from the 25. And what we can see here is that male heat stress reduces the probability of even presenting a gift. So these males are not going to mate because females aren't interested in them because they're not giving them any gifts. Even if they do give a gift, females are less willing to take them. But interestingly, the number of gifts presented is an interaction between the male and the female temperature. And we think what's going on here is stressed males are directing their attention to better conditioned females. So we might have a situation here where the operation of sexual selection is altering a little bit. Yes, they're sterile, but they're trying to get females to mate with them. Okay, so given that a male gives a gift, what does females do with it? So gift acceptance probability and gift acceptance latency, same structure, 18 males, 25 males, mating with 18 or 25 females. And male heat stress reduces both the probability of a female accepting a gift, and if she does accept it, she takes longer to accept it. So there's bias against mating with these heat stressed males. We have sexual selection potentially operating for male mate choice and for female mate choice. Okay, but what about the costs to offspring production? So remember, in that dietary stress, stressed females who mated with stressed males produced fewer progeny. Now, here what we're doing is we're plotting the number of offspring based on the number of gifts accepted for females that were mated female temperature. Yes, okay. So sorry, this is complicated. Now we're only looking at female temperature because we know that females that are mated to 25 males don't produce any progeny, right? So these are females that were treated at 18 or 25, but we're taking only the ones that mated to 18-degree males. Everybody with me? And here's the plots. And there's no significant difference between females that were raised at 18 and females that were raised at 18. But this relationship is predicated by the number of gifts accepted. And to reach that same level, these females accepted more gifts on average. So they can compensate for their core stress, but they have to make their males work harder and give them more gifts. Okay, so what we found with the subobscurate is we have genetic variation for heat-induced male sterility. We have genetic variation for, let's take that away, polyandry. And both of these things, we also have evidence for the gift giving that changes in sexual selection might be occurring dependent on male and female heat stress. And so this might alter other aspects of sexual selection. The fitness costs of mating with sterile males obviously results in reduced fitness. This includes a survival cost as well, and even following remating. And like I said, we have evidence of male and female mate choice changing under heat stress. Okay, so overall, you're nearly there. The ecological and evolutionary consequences of this thermal fertility limit. TFLs, better predictors than, well, they can be shifted to the left and occur before survival limits. TFLs better predict current species ranges and impact extinction probability, and that's going to result in more pessimistic forecasts, which I think is really underappreciated in thinking about how organisms are going to respond to climate change. It appears that there's little adaptive potential, but I think we need to engage in some experimental evolution work here to try and look at that more. But we identified some really good candidate genes that we can maybe do some comparative work on, for example. And finally, I think that heat stress might alter the mating system and subsequently affect the action of sexual selection, another downstream consequence on this. Okay, so those are the questions that we went through today. And I'd just like to thank all the FLY group and the STM group. And also, one of my questions was, does this happen in other organisms? Because it's so Drosophila-centric. Just going to give you a little taster. We started to doing similar stuff in butterflies, and the answer is butterflies are screwed as well. Okay, so thank you very much for your attention, and I look forward to any questions.